So this is the final part, part 4, and I'm going to talk about oxidative phosphorylation. Before I start talking about oxidative phosphorylation, it's important that we have a quick recap and remind ourselves of all the products that have been made to date. So glycolysis. At the end of glycolysis, we have produced 2 molecules of ATP. Remember, it was actually 4, but we used 2 to kickstart that stage. So it's a net gain of 2 that we have there. 2 pyruvate molecules, obviously, at the end, and then 2 molecules of reduced NAD. Whereas the link reaction, we will end up with 2 molecules of acetyl coenzyme A, 2 molecules of carbon dioxide, and also 2 coenzymes, 2 reduced NAD coenzymes. Now, remember that because 1 glucose molecule yields 2 pyruvate, that means that each of those pyruvate will go on in the link reaction to produce an acetyl coenzyme each, which is why you have 2 acetyl coenzymes. And to make an acetyl coenzyme from pyruvate, we lose the carbon dioxide and we lose the hydrogen. So that's why we've got 2 times carbon dioxide and 2 times hydrogen. So if you're looking back at stage 2 and thinking, but I can only see 1 acetyl coenzyme here in the link reaction, remember it happens twice because there's 2 pyruvate being formed as a result of glycolysis. Then we go to the Krebs cycle. So again, remember that there's going to be 2 Krebs cycles going on simultaneously for one molecule of glucose. Now, the Krebs cycle in total, there's going to be 4 carbon dioxide molecules produced. There's going to be 6 reduced NAD coenzymes, and there'll be 2 reduced FAD coenzymes, and also 2 ATP molecules. Now, it's the reduced NAD and the reduced FAD, they are really important in this next stage, which is called oxidative phosphorylation. Now, the oxidative phosphorylation is performed by the electron transport chain. So let's have a look at how that works. So if we zoom in first of all to the mitochondria, we know that the electron transport chain is embedded within the phospholipid bilayer of the inner membrane. So as we zoomed in there, you will notice that here's the inner membrane here, and we've got a series of proteins and an enzyme there. Here's your phospholipid bilayer. That part is the matrix, the fluid-filled area that you should know about inside the mitochondrion, and this is the intermembrane space that's between the inner membrane, and obviously you'd have the outer membrane there. So if you look back at the video where I actually built a model, the outer membrane is the red, the inner membrane is the blue, so the intermembrane space is obviously that void in between the two. Now, here's our reduced NAD. So here it comes. Remember, we're in the matrix to begin with, and that's obviously where the Krebs cycle happens. And let's say that this reduced NAD has obviously come from the Krebs cycle. Now, the reduced NAD is going to drop off the hydrogen, and that hydrogen, H2, is made up of 2 H atoms, and a H atom is made up of a proton and an electron. So this and this gives you a hydrogen atom, and then this and this will give you the other hydrogen atom, and then obviously combined it is a molecule of hydrogen, H2. So this is now NAD. It's dropped off that hydrogen, so it's lost a hydrogen, so it's now become oxidized. And that will go back to the Krebs cycle, or it could go back to the link reaction where it can then pick up another hydrogen and become reduced NAD. So remember, the coenzyme will take the hydrogen from one place to another. That's its job. So let's have a look what's happened to this electron here. So this electron has been dropped off at this protein here. And what will happen to that electron, that pair of electrons, is they will be passed from one proton to another in a series of redox reactions. So obviously as it gains, it becomes reduced. As it loses the pair of electrons, it becomes oxidized. And as a result of those pair of electrons moving from a higher energy level down the electron transport chain, they'll be losing energy, or they'll be releasing energy as they as the pair of electrons descend. And it's that energy that's going to be used to pump across those protons. So those protons that we saw that and obviously split off from the hydrogen, it's those protons that are going to be used to be actively pumped across this phospholipid bilayer. The only way that they can get across is obviously through these proteins here. This phospholipid bilayer is impermeable to protons, so they can't just move across. Anyway, what happens then is slowly you have a build-up of protons in the intermembrane space. So you're going to have a real high concentration in here compared to the matrix outside. That's another reason why the protons can't just diffuse across, is because they'll actually be going against the concentration gradient. So they need to be pumped in there. So that's what is happening there. And if we have a look at this next slide, because we've got obviously this concentration gradient, what these protons naturally want to do is they want to move from the intermembrane space here back into the matrix. But they can't. Like I said, this is impermeable to those protons. The only way they can actually get out is through this channel. It's called an ATP synthase channel. And what happens as they move through the ATP synthase channel, they cause a molecule of ADP and a molecule of inorganic phosphate to fuse together. And you can see that happening there. So this basically, them moving through there provides the energy that's needed to get that molecule of ATP to be formed. And then, because it's a conveyor belt, the electron transport chain is effectively a conveyor belt, the only way that another pair of electrons can start moving down the electron transport chain is if this moves on somewhere else. So we need something to accept this final pair of electrons here, and this is where oxygen is so important. So as you saw there, your oxygen, if I just move back again and I'll pause it, your oxygen is your final electron acceptor. That's why oxygen is so important in respiration. Without oxygen, then this whole conveyor belt will stop, because unless this pair of electrons is taken away, we won't be able to have another pair of electrons to start flowing from one protein to another. So a whole conveyor belt, the whole electron transport chain will stop working. So it's really important that we have oxygen. So this is the final electron acceptor. So what will happen, we can see that you've got your protons that have come through here, and those protons will combine with these electrons to give you an atom of hydrogen, and then two of those hydrogen atoms will combine with the oxygen, and therefore creating water, and that can be released. So you can see that happening there, and a molecule of water is released, and that is released out of the mitochondria, out of the cell, and that energy that is released as that leaves is enough to obviously carry over another proton into the intermembrane space. Now you can see that the electron transport chain is available to receive yet another pair of electrons. And this time, let's have a look at what FAD does. It does exactly the same, apart from the fact that instead of dropping off the pair of electrons at the start of the electron transport chain here, it'll drop them off slightly further up. And basically that means that, as a result of it, not as many protons will actually be actively pumped into the intermembrane space, because they don't have as much time. If the pair of electrons started off here, then obviously as they move from there to there, you'd have a proton move across there. But because they're starting here, there's not going to be as many protons that are going to be actively pumped in. And you can see that happening here. So the earliest that a proton can be pumped in is here, as the pair of electrons move from this protein to that protein there. It ultimately means that there's not going to be as many protons in the intermembrane space. And if there's not as many protons in the intermembrane space, there won't be as many protons moving through the ATP synthase channel. And if there's not as many protons moving through the ATP synthase channel, then there won't be as many molecules of ATP being formed. So that's why the coenzyme FAD, because of where they drop off the pair of electrons, it just means that they're not going to yield as many molecules of ATP after this this whole thing plays out. But in terms of what happens to the protons and what happens to the electrons, it's exactly the same as when we were looking at the NAD. So if we just quickly go back, have a quick look at that. So you've got your FAD is going to drop off its hydrogen. That hydrogen is going to be split up into electrons and protons, and then the electrons can move down the electron transport chain, and that's going to release energy so we can actively transport the protons across into the intermembrane space, and then they're eventually going to move out from the high concentration down the concentration gradient through the ATP synthase channel, and they're going to cause a molecule of ADP and inorganic phosphate to fuse. And then what's going to happen is the molecule of oxygen is going to come in, the oxygen atom, and that's going to be the final electron acceptor, and then it's obviously those electrons are going to combine with the protons to create a molecule of water, and that's going to pass out the mitochondria. And that is oxidative phosphorylation, and that's obviously going to happen time and time again, and they're going to be supplied with reduced NAD and reduced FAD as a result of the link reaction and the Krebs cycle. So hopefully you've understood that.